Hi dear students, this is Dr. Wilma from Bowring and Lady Curzon Medical College and Research Institute. This presentation is to facilitate you to understand one of the core competencies of protein and amino acid metabolism. At the end of this large group teaching, the students should be able to describe amino acid pool and also describe nitrogen balance. Define transamination with suitable examples. Mention the salient features of transamination. Explain deamination and transdeamination reactions with suitable examples. Explain the formation of biogenic amines with suitable examples and also enumerate the functions and clinical significance of biogenic amines. Define transmethylation and list any 4 transmethylation reactions. These are the specific learning objectives of this teaching session. Before going to the details of protein and amino acid metabolism, let us recapitulate the structure of an amino acid. Each amino acid consists of alpha carbon atom bonded by alpha amino group, alpha hydrogen atom, distinctive side chain, that is R group, and a carboxyl group. There are 20 amino acids which are commonly found as constituents of mammalian proteins. Out of these 20 amino acids, there are 10 essential amino acids which are also known as indispensable amino acids. They are histidine, threonine, tryptophan, phenylalanine, valine, isoleucine, leucine, lysine, methionine and arginine. So easy to remember it as HTP Wilma. So these are essential amino acids which has to be consumed in the diet, which cannot be synthesized in the body. Out of these 10 amino acids, the first letter H and the last letter A, these two constitutes the semi-essential amino acids. They are histidine and arginine. The amino acids which are synthesized within the body, which are not essential to be consumed in the diet, are known as non-essential or dispensable amino acids. They are glycine, alanine, serine, cysteine, aspartate, asparagine, glutamate, glutamine, tyrosine and proline. And the mnemonic for this is ACT, GASP, GAG. These essential and non-essential amino acids are very important to understand the concept of amino acid pool and the nitrogen balance. This amino acid pool is an imaginary pool which consists of free amino acids present throughout the body, such as in cells, blood and extracellular fluids. This pool is supplied by 3 sources. That's from the ingested protein, that is diet, as essential amino acids, and the synthesis of amino acids within the body as non-essential amino acids, and the amino acids which are derived from the tissue proteins. The utilization of amino acids from the amino acid pool directly for the formation of or the synthesis of purines, pyrimidines and porphyrins. And the degradation of amino acids releases nitrogen and the carbon skeleton. The nitrogen of the amino group of the amino acid detoxified to form urea and excreted from the body. And the carbon skeletons of ketogenic amino acid give rise to acetoacetate and acetyl-CoA. And the carbon skeleton of glucogenic amino acid give rise to pyruvate, alpha ketoglutarate, succinyl-CoA, fumarate and oxaloacetate. Muscle and liver are very important organs in maintaining this amino acid pool. Due to the maintenance of this pool, there is no immediate deficiency manifestation of proteins, and amino acids takes a prolonged persistence of protein deficiency for clinical symptoms to develop. Here is the fate of carbon skeleton of ketogenic and glucogenic amino acids. The ketogenic amino acids give rise to acetyl-CoA and acetyl-CoA, and the glucogenic amino acids like alanine, cysteine, glycine, serine, threonine, and tryptophan giving rise to pyruvate. And oxaloacetate is derived from asparagine and aspartate. Fumarate derived from aspartate and phenylalanine tyrosine. Succinyl CoA is derived from isoleucine, methionine, threonine, and valine. And alpha ketoglutarate from arginine, glutamate, glutamine, histidine, and proline. In healthy individuals, the input to the amino acid pool is balanced by the output. That is, the amount of amino acids contained in this pool is constant. The amino acid pool is said to be in a steady state, and the individual is said to be in nitrogen balance, which is nothing but nitrogen equilibrium. So when do you call it as a positive nitrogen balance? When the protein synthesis is more than the protein degradation, you call it as a positive nitrogen balance. And when the protein synthesis is less than the protein degradation, then you call it as a negative nitrogen balance. This is a state of nitrogen equilibrium where nitrogen input is equal to nitrogen output. This is a state of positive nitrogen balance wherein the nitrogen intake is more than the nitrogen excretion. It shows that nitrogen is retained in the body. This occurs in conditions like pregnant women and in infants. Here is a state of negative nitrogen balance wherein the tissue protein is utilized or the contributing for the amino acid pool. The conditions like stress or decreased intake. Here is a state of negative nitrogen balance seen in case of serious illness like advanced cancer or trauma, stress, or whenever there is a decreased intake, as in case of kwashiorkor, where the tissue protein is contributing to the amino acid pool and the nitrogen intake is less than the nitrogen excretion. Under general reactions of amino acids, transamination is one of the important reactions, and it's also a frequently asked question in the formative and summative assessments. So what is this transamination? Transamination is defined as the transfer of an amino group from an amino acid to a keto acid so that there is a formation of new keto acid corresponding to the original amino acid and a new amino acid corresponding to the original keto acid in presence of transaminases and the coenzyme pyridoxal phosphate. Here we can consider glutamic acid, which is an amino acid reacting with the keto acid, that is a pyruvic acid, so that the amino group from the glutamic acid is transferred to pyruvic acid or the pyruvate, so that the glutamic acid is converted to alpha ketoglutarate, so that the new keto acid corresponding to the original amino acid with the same number of carbon atoms and the original keto acid converting into a new amino acid, that is pyruvic acid, is converted into alanine without the liberation of ammonia and without the utilization of energy in the form of ATP. I would like to emphasize on the same reaction. Here an amino acid glutamate is considered and the keto acid pyruvate. So the amino group from the glutamate is transferred to the pyruvic acid, that is a keto acid, so that a new keto acid is formed, alpha ketoglutarate, corresponding to the original amino acid glutamate, and a new amino acid alanine is formed corresponding to the original keto acid pyruvate in presence of an aminotransferase, that is alanine aminotransferase, and a coenzyme pyridoxal phosphate. which is a reversible reaction without utilization of energy and without liberation of ammonia. Let us understand the role of pyridoxal phosphate in transamination. An amino acid, alanine, reacts with pyridoxal phosphate so that the amino group from alanine is transferred to pyridoxal phosphate and gets converted into pyruvic acid and the pyridoxamine phosphate. So the new keto acid is formed here, pyruvic acid corresponding to the original amino acid alanine, and the pyridoxal phosphate is converted into pyridoxamine phosphate after accepting the amino group. Pyridoxamine phosphate reacts with alpha ketoglutarate, that is the original keto acid, so that which accepts the amine group from the pyridoxamine phosphate to get converted into new amino acid, that is glutamic acid, and with the release of pyridoxal phosphate in presence of the transaminase. This is a structure of pyridoxal phosphate, which is a coenzyme of vitamin B6. Here we can see the highlighted structure, which is indicating that the aldehyde group of pyridoxal phosphate is very important for transamination reaction. This slide depicts the structure of an amino acid and an enzyme linked to pyridoxal phosphate by Schiff base. Schiff base is named after Hugo Schiff. It can be considered as a subclass of imines, being either aldimines or ketimines. In the resting state, the aldehyde group of pyridoxal phosphate, which is highlighted in yellow color, is in a Schiff base linkage to the epsilon amino group of lysine side chain of an enzyme. So we have an amino acid, free amino acid, and the enzyme linked to the pyridoxal phosphate by Schiff base. So when the reaction happens, the alpha amino group of an amino acid displaces the enzyme to form a Schiff base linkage to PLP. So here there is a Schiff base formed between an amino acid and a pyridoxal phosphate. The amino group remains on the pyridoxal phosphate as pyridoxamine phosphate, and an amino acid leaves as a new keto acid. A different keto acid reacts with pyridoxamine phosphate and the process reverses to complete the reaction. To summarize the transamination reaction, we have alanine aminotransferase and the pyridoxal phosphate, because alanine and the alpha ketoglutarate are utilized for the reaction as substrates. So the alanine reacts with pyridoxal phosphate to form pyruvic acid, the new keto acid pyruvic acid corresponding to the original amino acid alanine, and pyridoxal phosphate converted into pyridoxamine phosphate. Pyridoxamine phosphate reacts with alpha ketoglutarate to get converted into new amino acid corresponding to the original keto acid, that is alpha ketoglutarate, and pyridoxal phosphate is released. Here we can see the family of transaminases. For example, alanine reacting with alpha ketoglutarate to form pyruvate and glutamate. Hence the name of the transaminase is alanine aminotransferase. In most of the reactions here, we can see that alpha ketoglutarate accepting the amino group from the amino acids to get converted into glutamate, which is non-essential amino acid. This transamination reaction takes place in the cytoplasm of all the cells of the body. The amino group is transported to liver as glutamic acid. The salient features of transamination are transfer of an amino group from an amino acid to keto acid. Specific transaminases exist for each pair of amino acid and a keto acid. Pyridoxal phosphate is very essential for transamination reaction. As we have discussed that there is a formation of Schiff base, then the pyridoxal phosphate accepts the amino group from the amino acid and then transfers to the keto acid, and it is a reversible process without the liberation of ammonia, because there is only transfer of amino group between amino acid and the keto acid. Most amino acids are substrates for transamination except for lysine, threonine, proline, because these amino acids they undergo direct degradative pathway, hence they do not undergo transamination. Transamination is not restricted to alpha amino group only. For example, delta amino group of ornithine also undergoes transamination. Glutamate, which is a non-essential amino acid synthesized by transamination reaction by reaction of the alpha ketoglutarate with an amino acid, so that the alpha ketoglutarate gets converted into glutamate. In the same way, oxaloacetate reacts with amino acids to produce aspartic acid. And transamination diverts the excess amino acids towards energy generation, as well as amino acids undergoes transamination to finally concentrate nitrogen in the form of glutamate. And serum transaminases are also very important for diagnostic and prognostic purposes. The other general reaction of an amino acid is deamination, which means removal of amino group. So the removal of amino group from amino acids is known as deamination. It may be either oxidative type or non-oxidative type. The oxidative deamination may be by glutamate dehydrogenase or by amino acid oxidase. The non-oxidative deamination is by amino acid dehydratase. So the removal of amino group from amino acids is known as deamination. It may be either oxidative type or non-oxidative type. The oxidative deamination may be by glutamate dehydrogenase or by amino acid oxidase. The non-oxidative deamination is by amino acid dehydratase. The deamination reaction, which is taking place by glutamate dehydrogenase in presence of NAD plus and a molecule of water, converts glutamate to alpha ketoglutarate and ammonium ion. So the reaction is taking place in presence of NAD plus, hence it is known as oxidative deamination, wherein NAD plus acts as an oxidizing agent. Oxidative deamination by glutamate dehydrogenase requires either NAD plus or NADP plus as an oxidizing agents. This mitochondrial reaction is reversible, occurs in most tissues but mostly in liver. All amino acids are first transaminated to glutamate, which is then finally deaminated. Amino acids are deaminated at the rate of about 50 to 70 grams per day. This reaction is got a significant role because either it can be catabolic reaction or anabolic reaction. If it is a catabolic reaction, there is a formation of ammonia from glutamate by the removal of nitrogen. If it is anabolic reaction, then there is amination of alpha ketoglutarate to form glutamate. L-glutamate dehydrogenase can utilize either NAD plus or NADP plus. The utilization of two different coenzymes by glutamate dehydrogenase, that is NAD plus for release of ammonia and NADP plus for incorporation of ammonia, permits independent regulation of these two reactions. It is very important to note that independent regulation is possible although both the reactions are catalyzed by the same enzyme. The salient features of oxidative deamination are the liberation of free ammonia from the amino group of amino acids in presence of oxidizing agents, and this reaction takes place mostly in the liver and kidney. Here, the glutamate and the L-glutamate dehydrogenase serves as collection centers for amino groups to liberate ammonia. And this enzyme is very unique in its reaction because it can use either NAD plus or NADP plus as coenzyme, and increased level of L-glutamate dehydrogenase is usually seen in liver diseases. The other type of oxidative deamination by L-amino acid oxidase is to convert L-amino acid to L-amino acid in presence of auto-oxidizable flavins, that is FMN, nothing but flavin mononucleotide as coenzyme which oxidize amino acid to alpha amino acid. The alpha amino acid is an unstable compound which decomposes to the corresponding alpha keto acid with the release of ammonium ion. In this reaction, oxygen is reduced to hydrogen peroxide, which is later decomposed by catalase. The second type of deamination is by non-oxidative deamination by utilizing amino acid dehydratase, in which there is a liberation of free ammonia from the amino group of amino acid directly without utilizing the oxidizing agents. For example, serine getting converted into the keto acid pyruvate and ammonia is released in presence of serine dehydratase. In the same way, threonine getting converted into alpha ketobutyrate and ammonia in presence of threonine dehydratase. A functional coupling of two reactions, the transamination and oxidative deamination, is known as transdeamination. This is the major mechanism of removal of ammonia from the amino acids for detoxification and excretion as urea. The best example for the functional coupling of these two reactions, that is transamination and oxidative deamination, is conversion of alpha ketoglutarate to glutamate. Alpha ketoglutarate reacts with an amino acid in presence of transaminase and pyridoxal phosphate so that the keto acid is formed corresponding to the original amino acid and glutamate is formed from the alpha ketoglutarate. Glutamate, which is formed by transamination, undergoes oxidative deamination to release ammonia and alpha ketoglutarate. Alpha ketoglutarate, which is released, can be reutilized in the transamination reaction. The reactions of amino acids also includes the formation of biogenic amines by decarboxylation reaction. These biogenic amines have diverse functions and basic in nature. Removal of carbon dioxide from the carboxyl group of an amino acids results in the formation of amines known as biogenic amines. In presence of decarboxylase, the enzyme, and the coenzyme pyridoxal phosphate, we have several biogenic amines which are formed from decarboxylation reactions of amino acids. For example, gamma aminobutyric acid formed from glutamic acid, ethanolamine from serine, dopamine from dopa, tyramine from tyrosine, serotonin from pyhydroxytryptophan, histamine from histidine, putrescine from ornithine, cadaverine from lysine, taurine from cysteine. These biological amino acids have diverse biological functions. For example, gamma aminobutyric acid is an inhibitory neurotransmitter. In case of deficiency of vitamin B6, which is very much required for the formation of coenzyme pyridoxal phosphate, leads to decreased production of GABA, that is gamma aminobutyric acid, so that which in turn leads to convulsions in infants and children. Then we can consider histamine, which is formed from histidine, is produced by the decarboxylation reaction. It is a vasodilator and it lowers the blood pressure. It is involved in allergic reactions. So with this, we are going to complete the biogenic amines. Transfer of methyl group from methionine to an acceptor molecule so that the methylated product is formed is termed as transmethylation, and this transmethylation reaction is the last topic for today's presentation. The methyl group of methionine becomes available for transmethylation reaction only in an active form of methionine known as S-adenosylmethionine or SAM. How this SAM is synthesized? This SAM is synthesized from methionine in presence of ATP and methionine adenosyl transferase, where the adenosyl is accepted by methionine from ATP, so that methionine is converted into S-adenosyl methionine. Now, the methyl group from the S-adenosyl methionine is accepted by the acceptor, so that methylated products are formed and release of S-adenosyl homocysteine in presence of the enzyme methyl transferase. The methyl group of active methionine, that is SAM, is very reactive and can be enzymatically transferred in the synthesis of many compounds such as creatine from guanidoacetate, epinephrine from norepinephrine, melatonin from acetylserotonin, methylated tRNA bases from tRNA bases, and choline from ethanolamine. The methyl group of active methionine, that is SAM, S-adenosyl methionine, is very reactive and can be enzymatically transferred in the synthesis of many compounds such as guanidoacetate getting methylated to form creatine, norepinephrine converted to epinephrine by methylation, acetylserotonin to melatonin, and methylated tRNA bases, ethanolamine getting converted into choline, so that S-adenosyl methionine after methylation are donating the methyl group getting converted into S-adenosyl homocysteine. Transmethylation reactions are very important for the biosynthesis of number of biological products. As indicated in this table, to name a few, we have epinephrine, choline, melatonin, methylhistidine, etc. With this, I would like to conclude this competency. Thank you all, dear students.